We're talking about some crazy deep sea jellies. They caught some of these. They were in the jar and then they just vanished. The amount of pressure that these guys are dealing with is just bonkers.
SciAnts. We get experimental. SciAnts. We're curious, non-judgmental.
Okay, so I want to talk to you guys about comb jellies. But first, let's talk about jellies in general. We're going to turn to Embari for a minute.
Yeah.
Okay, normally when we talk about jellyfish, we're talking about the cnidarians, but that's not what we're talking about tonight. We're talking about comb
They all have
jellies.
in common the presence of...
Yeah. So we were when we were down in Florida, we caught a bunch of comb jellies.
And we got to hold them in our hands.
It was wild. It was crazy. And they don't have the stinging cells, which is one of the reasons. And also, the guys we were catching were pretty robust, and we were careful. So we didn't hurt them. They didn't hurt us. Everyone went their separate ways, reasonably happy.
I did not get the memo when the young woman was reaching in and grabbing them that they don't sting. And so I just thought she was a wizard, like going in with her bare hands, like, oh my god, what are you doing?
What do they feel like? Oh, I gotta be honest, kind of felt like holding a glob of Jell-O in water. A somewhat sturdy glob of Jell-O inside a pan of water, Balint. Like, it's just as far as, like, the texture and the delicateness of it, I mean, I know it can hold up more. Like, it had that texture and the sort of wobbliness. It is more structurally sound than that, but just, like, if you want the sensation. Yeah. All right. So, ctenophores, the comb jellies. There are a lot of comb jellies. There's over 180. I want to say it's like 184 or something like that species of comb jelly. So there's a huge variety of ctenophores. Some of them are known for being super deep ocean, which is what we're talking about tonight. The ctenophores, the comb jellies. But we're talking about some crazy deep sea jellies. 5 kilometers deep. All right. Scientists have been wanting to study them for a long time. It was a couple of years ago when they were in a manned submersible. They caught some of these really, really, really deep sea jellies. They caught them to bring them back up to the surface so that they could study them. And on the way up, they disappeared. They were in the container, they were in the jar, and then they just vanished. And they thought, oh gosh, something terrible went wrong. So, and there's like a big temperature gradient. The really deep ocean is just barely above freezing a lot of times. So they thought, we really stressed these guys out. They must have disintegrated or something. A couple years later, they went back and they tried to do like a temperature controlled housing unit for them and brought them up, and they disappeared again. They were disintegrating because of the pressure change. So much of their physiology is designed to accommodate that extreme pressure that they literally fall apart at normal atmospheric pressure. Anyway, so this paper that came out, scientists figured out what it is about these jellies that allows them to live in the extreme deep, and kind of the mechanics, like the biophysics of the molecules that allow them to live in the deep but to disintegrate at atmospheric temperature.
Maybe you'll get into it a little bit. So is it like at the cellular level?
Yeah, it's at the subcellular level. Like, I'll give you guys spoilers here. Each of our cells is a unique little unit, and it has a membrane coating on the outside. That's what keeps the stuff inside the cell inside, and the outside is the outside. There's a membrane layer, and a lot of that is made up of these it's lipids mostly. Other stuff too. It's called a lipid bilayer, but anyway. So their lipids, the stuff that actually makes the structure of the outside of the cell, is really different than other jellies. I mean, other animals, but even other jellies that are closely related, but found closer up to the surface. Yeah, so it's actually at the supramolecular level. It's the stuff that builds the membrane around each cell is really different, has different mechanical properties. They later, in the paper, they actually found that, yes, like the membranes like fall apart and form these little tiny itty bitty tube structures, but it can't see it by the naked eye. They just disintegrated. Freaking vaporized. Practically vaporized. Jellyfish in a jar. These jellies live 5 kilometers, miles. The amount of pressure that these guys are dealing with is just bonkers. Okay, so this is what happens in the human body after just a little while. But yes, chemistry starts to change. The amount of the kinds of gases that can be dissolved, the ratio of gases that can dissolve in fluid like your blood changes. The amount of pressure underwater is very different than the kind of pressure change we experience when we go up or down in elevation. It's because water is so much heavier than the gases that make up our atmosphere. And when you're dealing with things under that kind of pressure, chemistry actually starts, like the rules of chemistry at atmosphere level start to change. Pressure does some bonkers stuff. And so animals that are adapted to the really deep oceans, have to come up with some clever stuff. And it shouldn't be shocking to us that they don't do too well up at the surface. Okay.
Yeah, you're totally right. It shouldn't be shocking.
It shouldn't be shocking.
But it's like collapsed.
Yeah. It is still really cool. Like, how do freaking whales do deep dives?
Or I guess because you'll be talking about sharks at some point, but the ones that dive down and then come upward to get their prey, those two, I mean, those are
sharks don't tend to dive as deep as
the whales.
Whales, though. I mean, because the sharks do attack from underneath, but they don't do the same level of traveling that a lot of the whales do for, like, their deep dives. So animals have amazing physiology to get around this. Us squishy humans, we weren't meant to be in the deep, so we don't have those adaptations. So, our lungs collapse and blood chemistry gets all crazy. However, these jellies are freaking bonkers. So the trick to your cell membranes, right? So the little thin layer that keeps, imagine like a balloon, all right? And your cell is a balloon because it's 3-dimensional, okay? The little rubber exterior is this membrane we're talking about. So it's just what separates the inside, all of your good inside cellular juices from the outside extracellular juices. That membrane is made up of these fatty acids. It's made up of a lot of stuff, but it's made up of these fatty acids. And the trick to these membranes being successful is that they're not rigid. Your cell membranes are not rigid. Balint, your cell membranes are not rigid. Our cell membranes are not rigid. They can't be. You can't see it. Even under a normal microscope, you can't see this. But on an incredibly fine scale, there's a tiny bit of movement. We need gas exchange back and forth. We need things to, in like controlled ways, be able to get through this membrane. That's how all of the chemistry has worked. So, I mean, we want it to be rigid enough that it's a structure, right? Think about the balloon. We need the rubber balloon to still be a balloon, but we can't have it be rigid. If it gets really, really rigid, the cell's just gonna die. And it turns out that keeping that structure is really hard. Keeping that sort of flexibility is really hard at really deep levels. And so the lipids that they have, the molecules that make up this membrane, are these really big, crazy, they kind of make like a V shape under low pressure. They have a huge top and a little skinny bottom. And so under low pressure, that you can't make a nice layer out of that because everything's all wonky shaped. But under extreme pressure, they kind of zip up and they don't become perfectly narrow. We don't want everything narrow like this. That's getting rigid. But they keep the shape that our membranes have at sort of our, 1 atmosphere of pressure. Way down deep, they're able to take on a more predictable shape. And that's why. So that's the key to this jelly's physiology, is it just has a different fatty acid. It has a different chemical molecule that's making up this membrane. And it looks like ours, only at really deep pressure. All right. And so one of the ways they did this, which was really cool, is they took some bacteria and they were like, I don't know, let's see what happens if we mess with like bacteria's lipids. So they changed the membrane on bacteria and put it under extreme pressure. And when they swapped out what the jellyfish has and put it in the bacteria was able to withstand this insane amount of pressure also.
Oh, that's really cool.
Yeah. Right.
Wombat's wondering if it's like an accordion-like structure.
Not quite, but I get what you mean. So it does, yeah, like it, under pressure, yes, it does. I don't think the structure is exactly accordion-like, but it's the same idea. Like under pressure, you can kind of narrow it. And it needs that certain shape. It only keeps that certain shape under pressure. And it's just under crazy pressure.